Myself, Mr. H.T. Mate, Department of Botany, RBNB College, Sri Rampur. Today, we are going to study the F.Y.B.Sc Botany from this semester second and paper first BO 121. And the name of the paper is Plant Life and Utilization, and from this, the topic number 2, that is the Pteridophytes. And in the Pteridophyte, there is the life cycle of the Nephrolepis. So, the reproduction, it reproduces asexually, sexually and vegetatively also. So, first that is the asexual reproduction. Now, the plants are a sporophyte. The plants of the Nephrolepis is a sporophyte, and it produces the asexually reproductive structures. They are called as the spores. Now, these are spores which are asexual in nature. They are developed on the leaflet. That means on the leaflet, some dotted structures are developed. They are called as the sori. And in the sori, there are very large number of the sporangia. And in each sporangia, there are large number of the spores. So, these are the asexual reproductive structures. Now, they are developed on the leaflet. So, this is about the position of a sori on a leaflet. Now, these are sori or sorus. They are developed from lower surface of the leaflet. So, this is the diagram of the lower surface of the leaflet. It has midrib at the center, and there are the lateral veins. They are forked. And at the vein ending, some dotted structures are get developed, and they are called as the sori. So, this is from the lower surface of the leaflet or from the pinnae. So, this is about the position. Now, this is the outline of the sori. Now, this is the placenta, and on this placenta, very large number of the sporangia are get developed. So, this is the placenta and the sporangia. Now, this is the TS of the leaflet or pinna passing through the sori. So, this is the TS of the leaf. So, this is upper epidermis, this is lower epidermis, and in between these, there is the mesophyll tissue. And just at the vein ending, there is a development of the sori takes place. Now, it has a complicated structure. There is a protective covering, which is called as the indusium, which is from the lower surface. And in between this indusium and lower epidermis, there are the sori or the sporangia are get developed. So, this is the placenta. They are parenchymatous in nature. So, these are the placenta. They are parenchymatous in nature. That means they provide the food material to the developing sporogenous tissue. And at the end of a placenta, there is a very short stalk of the sporangia, and above this, there are club-shaped sporangia are developed. So, this is the TS of a leaflet or pinna through the sorus. Now, there is a indusium is there, placenta is there, and a number of sporangia are also there. Now, this is the detailed structure of the sporangia. Now, this structure is get divided into the two parts. The lower part, which is the few-celled, is called as the stalk, and above this there is the capsule. That means the body of the sporangia. Now, it is somewhat thumb-shaped in outline. It has a protective covering, which is called as the annulus. Now, the walls of the annulus, that is the inner wall and radial wall, are very, very thick, and only towards one side there are very thin walled cells, and they are called as the stomium. And inside both of these walls, there is a very large number of cells are developed, and they are called as the sporogenous tissue. So, this placenta provides the food material to this developing sporogenous tissue. So, in this sporangia, there is the sporogenous tissue, which is more important. Now, this sporogenous tissue acts as a spore mother cell. So, this is the spore mother cell. Initially, it is diploid. Now, this spore mother cell divides meiotically into a spore tetrad. So, this is the spore tetrad. So, these are the 4 cells. Tetrad, that means these are the 4 cells, 3 from the front side and 1 from the back side. And they are formed after meiosis. So, they are haploid in nature. So, this is the spore mother cell, which is also called as the sporogenous tissue. Now, it acts as a spore mother cell because it is going to divide, and it divides meiotically into a spore tetrad, and in each tetrad there are the 4 cells. All are haploid in nature because they are developed after meiosis. Now, this is the spore tetrad, and in each spore tetrad there are the 4 haploid spores. Now, they are called as the spores. Now, the structure of the spores, they are somewhat circular in outline at maturity. And each and every spore has 2 wall layers. The outer wall layer is very thick, it is sculptured and it is called as the exosporium. While inner wall is very thin, it is delicate and it is called as the endosporium. Now this is about the structure of this spore which is haploid in nature. Now next point, that is the dehiscence of the sporangium. Now when there is a dry weather, in this condition, this annulus, cells of the annulus and stomium dries up, and at a stomium where the cells are very thin, they are get ruptured. But before this, all the spore mother cells get developed into the spore and these spores becomes mature, and at this condition there is a rupture of stomium takes place, and with the help of wind all of these spores get released in the atmosphere. So these spores, they falls on a suitable substratum, that is on the wet soil, and then they start to germinate, and then there is a development of the next generation which is called as the gametophyte.